Welcome back, my dear students, in the new class. Today, we shall deal with the final segment of the centromeric structure and function, and we shall see that how the centromere maintains the segregation fidelity of chromosome while the cell is dividing. In the last few classes, we have dealt with the structure of centromere, the specification of centromere by attachment of centromeric protein A, and then the epigenetic specification of centromere by methylation of various sites in the centromeric DNA as well as the pericentromeric DNA. So let's begin with the slide share. So the last slide was concerned with the loading of CNPA during the cell cycle. In the concluding section of the last class, we have dealt with that the centromere specifying protein CNPA is synthesized during the cell cycle quite early in the human beings. So most of the proteins are synthesized during the S phase of the cell cycle, whereas CNPA is synthesized between the NA phase and G1, end of the G1 phase of the cell cycle. And so here you see that in this phase, the CNPA assembly is taking place. Now, this CNPA is doubled, that is, its replication takes place. And so we have, with the DNA replicated here, you have got the single copy. And when the DNA is replicated, it will have the two copies, two sister chromatids, and they will proceed. These two, you see that they are having CNPA as well as the canonical nucleosomes. So during mitotic phase, again, these two will drift to the two different poles, and again, the CNPA assembly in the DNA will take place. CNP synthesis will take place at the G1 phase itself, and the CNPA assembly will occur while the chromosome, while the sister chromatid, one of the two sister chromatids is passing through the G1 phase. So it is quite conspicuous that the synthesis of CNPA and assembly of CNPA nucleosome take place at very early in human beings during the cell cycle, and most of the proteins are synthesized during the S phase, whereas it had been synthesized early. So in the most phases of this, in the almost all phases, it keeps its maintenance because it had been synthesized quite early during the cell cycle. Here, DNA replication has taken place and the chromatin assembly has taken place, and now it is entering the M phase. So although timing of the CNPA loading varies across the different species, in contrast to canonical chromatins, CNPA nucleosome replenishment in higher eukaryotes occurs independently of the DNA replication. DNA replication is taking place here, but replenishment has already occurred before the replication. In animals such as humans and Drosophila melanogaster, CNPA is loaded between NA phase and here was the end of the M phase, anaphase, prophase, metaphase, anaphase, and telophase. So here from the end of the, let me have the pointer. So here, during the latter phases or during the terminal phases, in between the terminal phase of the cell division and the end of the G1 phase, it has taken place, its synthesis has taken place. DNA replication independent assembly is specific to CNPA nucleosome, ensuring its essential presence at centromeres throughout the cell cycle. Sorry. If synthesis would have taken place here, this phase of the cell cycle might have been without the centromeric protein A, but since it had been synthesized at the very outset, the entire cell cycle is having the centromeric protein A, ensuring its essential presence at centromere throughout the cell cycle. Centromere is present throughout the cell cycle. Centromere identity is maintained in the cell that exit the cell cycle for longer periods. I have already dealt with that. Many mammalian, in case of mammalian oocytes, the development of oocyte takes place while the embryo is still in the womb, and the ultimate or the final development takes place at the time of ovulation. So there is a gap of decades, and even during this quiescent phase, you can say the suspended cell cycle phase, or you can say that the cell has come out of the cell cycle, the centromere is maintained. Now, the ultimate goal of having centromere is that we should have segregation of chromosome in a fiddle way, in a faithful way, in a precise way, so that the two resulting daughter cells should have the same complement, full complement of the chromosomes. The chromosomes are in any one of the two must not be lost in some way, or a part of chromosome must lost in any way, and because that will have catastrophic consequences for the daughter cell, and it will lead to many anomalies such as aneuploidy, cancerous development, and all that. So there are 2 or 3 checkpoints. These checkpoints emanate from the point of attachment of centromere and kinetochore. So it is not, I have said earlier too, that it is not that the centromere is a passive docking point of the kinetochore. Rather, the centromere and kinetochore complex acts as a very much active and alert, vibrant point, a safeguard to maintain the fidelity of the chromosome. So it keeps an eye on the event which is leading to the segregation of chromosomes to the 2 different poles. So a spindle assembly checkpoint is crucial to maintain stability, since it prevents premature separation of the sister chromatids. So a spindle assembly checkpoint is an arrangement which prevents the segregation of chromosomes before they have been arranged bi-orientationally, that is, they have been arranged on the equator facing the 2 different spindle tubules, and those tubules have been attached to the centromeres. If those tubules are not attached to the centromere kinetochore complex will not feel tension, because after the attachment of the microtubule or spindle fibrase, the spindle fibrase will tend to move in the opposite direction, and it will cause tension at the point of the attachment, that is the centromere. So when the centromere is not attached to the spindle fiber, it will not feel the tension. And since it will not feel the tension, the spindle assembly checkpoint will be activated. And this is a spindle assembly activation of a spindle assembly checkpoint will lead to the formation of mitotic checkpoint complex. And this mitotic checkpoint complex will inhibit anaphase promoting complex. Anaphase promoting complex will promote the segregation of two sister chromatids, and this segregation will be initiated by dissolution of the attachment point and then moving of those sister chromatids towards the opposite poles. So when the sister chromatids or the centromeres have not been attached on either side of the when centromeres have not been attached by the spindle fibers emanating from the centrosome complex on either side of the chromosome, it will not feel tension. And if tension is not felt, then a spindle assembly complex, spindle assembly checkpoint, SAC, is activated. So SAC, which is activated by lack of tension at the centromere kinetochore complex, is the crucial stage for maintaining the segregation fidelity. SAC is crucial to maintain genomic stability, since it prevents premature separation of sister chromatids in mitosis and ensures the fidelity of chromosome segregation. So we were discussing, we have made all these discussions to ultimately ensure the fidelity of segregation of chromosomes. So entire development of centromere, its epigenetic specifications, and finally its attachment to the spindle fibrase or microtubules spindle fibrase, the entire function is to result in the segregation of chromosomes in a fidel way, in a faithful way, so that the two poles could have the complete complement of the chromosomes. So here, prevents premature separation, SAC, prevents premature separation of the chromatids, and it ensures the fidelity of chromosomal segregation. The SAC arrests the cell in mitosis until all kinetochores are stably attached to the mitotic spindle. So if kinetochores are not attached to the mitotic spindle, the kinetochore complex will not feel the tension, and since tension will not be felt, it will lead to the activation of spindle assembly checkpoint. The lack of tension will be felt by spindle assembly checkpoint. And if the tension is not being felt by this point, it will lead to the activation of mitotic checkpoint complex. Okay, so SAC arrests, it arrests the cell until all kinetochores are attached to the mitotic spindle. How it feels that all the kinetochores have not been attached to the mitotic spindle? By not feeling the tension. So what happens? How it arrests? Improperly attached kinetochores activate SAC, spindle assembly complex, and catalyzes the formation of mitotic checkpoint complex. So here another complex comes into the picture. If SAC feels that all the kinetochores have not been attached to the microtubules, it will arrest the mitotic process and it will activate the ACC and catalyze the formation of MMC. So ACC will be activated and it will catalyze the formation of mitotic checkpoint complex. The latter acts as the effector of ACC. That is, it brings into effect the goal that SSC was trying to accomplish. So it acts as the effector of the ACC. The MMC binds and inhibits anaphase promoting complex. Here is another complex which is called anaphase promoting complex cyclosome, to which this MMC binds and it inhibits E3 ubiquitin ligase. It also helps in the segregation of chromosomes. So this binding will stall the process of segregation of chromosomes. So let me relate it once again for your purpose, for the purpose of your understanding, that we have three complexes here. A spindle assembly checkpoint, one, then MMC mitotic checkpoint complex, another point, and then anaphase promoting complex. So coordination between these 3 points leads to the segregation fidelity. Now, suppose that all the sister chromatids which have been arranged at the equator, they are not attached to the spindle microtubules, or they have not been attached to the equator at all. So what happens that if they are not attached, the point of attachment, that is the kinetochore centromere complex, it does not feel the tension of attachment. The attachment itself does not create tension, but the attachment leads to the separation, leads to the pulling apart, and so a tension is felt. So if the attachment is not perfect, the tension will not be formed, and then a spindle assembly checkpoint will come into picture, and it will lead to the formation of mitotic checkpoint complex, which acts as the effector of a spindle assembly complex. So the ultimate goal of the spindle assembly complex is to arrest the process of segregation of the cell division. And so this goal is achieved by MCC. And so you can say that MCC is bringing into effect the target, the goal of ACC, and in that way it is effector of a spindle assembly complex. The MMC binds and thereby inhibits. Now, the MMC, what does it do? It binds to the anaphase promoting complex cyclosome. Anaphase promoting complex promotes the segregation of sister chromatids. So if the binding is not proper, a spindle assembly checkpoint will come into picture. It will arrest the segregation by mitotic checkpoint complex inhibiting the anaphase promoting complex. So anaphase promoting complex, if it is inhibited, the sister chromatids will not proceed towards the anaphase, and it not only inhibits the anaphase promoting complex, but also inhibits the E3 ubiquitin ligase, and this ligase, inhibition of the ligase, will also inhibit the movement apart. Once SS is satisfied, now after the arrest, the attachment process will take place. The sister chromatids or the kinetochores will have ample time to rearrange, to get bidirectionally arranged, orientationally arranged at the equator of the cell, and then the microtubules will come to attach to the centromere kinetochore complex. And if the attachment process had been perfected, then APCC anaphase promoting complex is promptly activated, and it targets the cyclin B, securing the degradation of cohesin, the cohesin enzyme, which we have seen that the pericentric chromatin, methylation of pericentric chromatin, promotes the incorporation of cohesin. So the cohesin will be targeted, securing for degradation of the cohesin. Cohesin will be degraded, and the two sister chromatids that were up till this point attached at the point of the centromere will get segregated with the cohesion, with the degradation of cohesin, and gradually the tensile will pull them apart towards the poles. So we can visualize this. So now you can see here that at this stage, you can see that the bivalent chromosomes, they have not been properly bio-orientationally arranged along the equator. And due to this misarrangement, the spindle fibers of this pole has got attached, but the spindle fibers of this pole has not got attached to the centromere of this chromosome. So it has been attached only one-sided. And if the cell division progresses, what will happen? Both the sister chromatids will pass to the one cell. So this daughter cell will have the two sister chromatids, while this daughter cell will have none. So the progress of cell division at this stage will be fatal, will have very dangerous consequences, will have fatal consequences. It will lead to aneuploidy and development of cancerous cells. So the cell division should be stalled. Okay. So you see here that this kinetochore, which has been attached to the centromere, is not feeling the tension of the attachment of the spindle fiber. If had the spindle fiber attached bidirectionally, it would have felt pressure from both the poles. But if it has not got attached by this side, it will easily move and no pressure will be felt here. So the attachment, improper attachment, has led to lack of tension at this point. So tensionless kinetochore is due to unattachment. Now it activates mitotic checkpoint complex. So this tensionless kinetochore, tensionless state of kinetochore, it being formed by this spindle, is being sensed by the spindle assembly complex. It arrests cell division by activating the mitotic checkpoint complex, and this mitotic checkpoint complex is inhibiting the anaphase promoting complex. So the progression of cell division will occur only when this anaphase promoting complex is activated. So here you see that tensionless kinetochore is being sensed by the spindle assembly complex, and the spindle assembly complex, feeling the lack of tension, will activate the mitotic checkpoint complex, MCC, mitotic checkpoint complex. It has been made up of several protein units. And so it stalls, it inhibits the APC and stalls the process of cell division. Where at this point where the sister chromatids or bivalent chromosomes have not been properly attacked. But in this case, you see that upon the installment of the cell division, they have got ample time to get arranged properly bi-orientationally at the equator and have got attached to the microtubules of the spindle on either side of the centromere, this side too and this side too. So the kinetochore here will feel tension from the either side, and it will lead to the inhibition of this mitotic checkpoint complex, and this mitotic checkpoint complex inhibition will lead to the activation of APCC, that is anaphase chromatic complex cyclosome. So what happened when chromosomes got arranged along the equator and got attached to the spindle fibers on either side of the chromosome? This mitotic checkpoint complex got inactivated, and that inactivation led to the activation of APCC, anaphase promoting complex, and this anaphase promoting complex has triggered the segregation of chromosomes, and you can see the sister chromatids have been segregated in equal halves. Entire complement will be made available to this cell and to this cell also. So the two daughter cells will have the entire complement of chromatids, and there is no question of any aneuploidy creeping in or any anomaly creeping in. So that's how the segregation is maintained. So let's see the next slide. What is happening? Here, the segregation attachment is not proper. This chromosome is attached only on this side and this on this side, and so the tension is not being felt at this point. And so due to the lack of tension, here the mitotic checkpoint complex have been activated, and this mitotic checkpoint complex activation has led to the inhibition of anaphase chromatic cyclosome complex, and this inhibition had stalled the cell division here. But when they have got arranged properly, bi-orientationally arranged, so that the spindle fibrous microtubules from either side can have the reach to the centromeres, they got attached to the centromeres from either side. That this point, kinetochore centromere complex is feeling the tension, and so this tension felt by the kinetochore has led to the inactivation of mitotic checkpoint complex assembled. The assembly complex, spindle assembly complex, has felt that tension is being maintained on either side, and so it has inactivated the mitotic checkpoint complex, and this mitotic checkpoint complex inactivation led to the activation of anaphase promoting complex, and that anaphase promoting complex asked the cell to proceed further, and it led to the proper segregation of the sister chromatids to the either poles, and that's how the fidelity of the chromosome is maintained while the cell division is going on. I have tried to simplify the entire process in an in profiles diagram. Here you see that the same diagram had been simplified here, that spindle fibers are have not been attached properly, and so misaligned chromosomes, spindle assembly complex senses, and here the MMC have been omitted. So the segregation is off. When chromosomes are bi-orientationally arranged, that is, they are arranged on the equator in such a way that spindle fibers from both the sides can have the reach to the centromere, then this APC, anaphase promoting complex, had been activated and the segregation process had begun. So I have tried to simplify the entire process in a few steps. If the chromosomes are misaligned, if the centromeres are misaligned, chromosomes are misaligned at the point of centromeres, the point of attachment, misalignment of chromosome means, what does it mean? Alignment takes place at the point of kinetochore, and kinetochore is attached to the centromere. So this misalignment of chromosomes means the unattachment of spindle microtubules to the centromere kinetochore complex. So if the chromosomes are misaligned, as in this case, what will happen? Activation of a spindle assembly complex will take place, and this spindle assembly complex via MMC, as we have seen in this diagram, via MMC, spindle assembly complex had felt that the tension is not being built up there, and so it has inactivated APC via this MMC. So here you see that the spindle assembly complex has been activated, and its effector is the MMC, and it causes inhibition of anaphase promoting complex. Here it is anaphase promoting complex. So misalignment leads to the activation. So here misalignment had led to the putting on of SAC, spindle assembly complex, and it led to the activation of MMC, and this MMC was inhibition of APC, and this inhibition of APC led to the stop signal that cell division will not take place here. So it will stop the further segregation of chromosomes until the chromosomes are properly arranged bi-orientationally and they got attached to the centromere on either side of the centromere kinetochore complex. So this is the inhibition state, stop signal is emanating, but when bi-orientation of chromosomes takes place, as in this case, what will happen? Inactivation of ACC, spindle assembly complex, will be inactivated, and it will, this inactivation of a spindle assembly complex will not lead to the MMC formation, and so the inhibiting impact on the APC will be withdrawn, and APC, that is anaphase promoting complex, will activated. As here it has been activated due to the proper bi-orientational arrangement of chromosomes and attachment of spindle fibers on either side, and this signals that everything is okay, and so the SAC is not activated because it is feeling the tension on either side, and it is getting all okay signal by the tension, and so MMC does not come into picture, and APC, the anaphase promoting complex, is activated and the cell division proceeds. So that's how the segregation of chromosomes in a proper way is maintained, which in other words we call the fidelity of segregation is maintained. So ultimate goal of the centromere kinetochore attachment, proper attachment, formation of complex of centromere kinetochore, and then attachment of the microtubules of the spindle fibers, where was to segregate the chromosomes on either side in a proper way so that entire complement of chromosomes are available to each of the sister chromatids, so that each of the daughter cells, so that the daughter cell can develop in a proper way. And if any part of the chromosome or a particular chromosome is lost, then it will be genetically impaired. It will lead to the aneuploidy, and it will have fatal consequences for that particular cell. And at long last, we can conclude by this diagram. Here you see that centromere, and this centromeric DNA. On this centromeric DNA, the proteins such as CNPA and constitutive centromeric protein network. So it includes various types of proteins. So centromere proteins have been attached at the DNA, and to this complex, kinetochore has got attached. So enlarging this junction, we can see a picture like this. Now the chromosome segregation is taking place. So spindle is attached at this point of chromosome, and it is segregating the chromosome towards the pole. As we have seen that in between the end of the M phase and end of the G1 phase. So between NA phase and end of the G1 phase, the centromere protein CNPA is synthesized, and the CNPA nucleosomes, centromeric nucleosomes, come into existence. And so you see that at the very outset, this centromere, this chromatid has got the centromere, and now when it when it passes to the S phase, the replication takes place, and here you see that the replication fork, that is the segregation of unfolding, uncoiling of the two DNA helices, and then replication is taking place. So replication fork, Y-shaped replication fork has been formed, and so here replication process is taking place. And now finally, coming to the G phase, you can see that the two sister chromatids have been completely replicated, and they have come into existence with a point of attachment, which is denoted by the centromere. And now kinetochore assembled on that centromere. So here, this is the DNA of the centromere on which proteins have assembled, and now upon it, kinetochore gets assembled. So this alloys structure is the kinetochore, and now the bivalent chromosome is fit for entering into the M phase, and after entering into the M phase, it has got the entire structure for the attachment of the spindle fibers, and if it gets arranged bi-orientationally at the equator, the spindle microtubules from either side will get attached to the centromere on either side of the bivalent chromosome, and the sister chromatids will be pulled apart in their entirety. That is, no sister chromatid will be pulled in such a fashion that both the sister chromatids could go to the one cell or the other daughter cell. So that's how the segregation of fidelity is maintained, and that was the purpose of centromere kinetochore complex. So that's how the centromere kinetochore complex performs its function for maintaining the segregation facility, and this segregation fidelity, and this fidel segregation will lead to each of the daughter cells having complete set of chromosomes, on either side, and they will develop properly. And that's how we end to our session, which was concerned with the centromere and chromosomal fidelity, segregation of the chromosomal fidelity. So in this prolonged session, we have seen that how centromere is developed. What is the centromere? It is point of attachment between the two sister chromatids at the primary construction, to which the kinetochore is attached. And how it is specified? It is specified by the attachment of centromeric protein A or its variants at that point, at that centromeric DNA. And the CNPA is a variant of canonical histone H3. It differs from histone H3 in having different N terminals, and there is a CATD point in the characteristic histone fold, which differs up to 40%. About 22 amino acids differ from the canonical H3, and that's how it differs from the canonical H3. And at the centromere, it pairs with, or it gets complex with the H4, and sen H3 H4 complex is tightly bound, and that sen H3 H4 complex form tetramer, to which two dimers of H1, H2A, H2B are attached, and that's how the octamer comes into existence. We have seen in detail by comparing the diagrams that the canonical nucleosome differs from sen H3 nucleosome in that the latter being more compact and occupying lesser area, and so they wrap about 120 nucleotides around them, whereas the canonical nucleosome wraps about 146 nucleotides into 1.65 turns around them. So that's the main difference. And this compaction leads to the two centromeric nucleosome coming in close contact with each other, and this close contact leads to the aggregation of centromeric nucleosomes more closely, and this closer aggregation will lead to the imparting physical strength to the point of the centromere, and it is necessary, it should be physically strong, its integrity should be maintained, because it will have to face the pulls from either side during the NFA while the sister chromatids are moving towards the two poles. So that's how its integrity is maintained. Now, the wrapping has also special features. The wrapping is slightly loosened in case of sen H3, and this loosened wrapping leads to the entry of, it facilitates the entry of this, and those entry of proteins are necessary for centromeric function. Sorry. Coming to the part 2, in the first section of the part 2, we studied about the different types of centromeres, and beginning from the point centromeres of the budding yeast to the original centromeres of the fission yeast, and to the more complex satellite regional centromeres of the human beings and other animals. So these different types, they are structurally different. They occupy more space on the chromosome, and you can correlate the point centromere to the original centromere too. So in the original centromere, the expanse of the nucleotide at the centromeric point has been increased, and this increased expand leads to the incorporation of more proteins, more CNP8 and other nucleosomes, and so there are multiple points of attachment for the spindle fibers per centromere, whereas in the point there was only one point of attachment. Coming to the human beings, that we have seen the satellite DNAs, and we have seen that how satellite DNAs are tandemly repeated segments, and how AT-rich satellite DNAs, when they are subjected to centrifugation, they occupy a separate band different from the main band, and that's how they are called satellite DNA. So GC-rich satellite DNAs can be heavier than AT-rich. Now, coming to the organization of the satellite DNA in the human centromere, we have seen that the repeated family is of alpha centromere, alpha satellite DNA family, in which the monomers are made of 171 amino acids, 171 nucleotides. So 171 nucleotides are the monomers which are replicated, and they are hierarchically arranged to get higher order repeats. That is, many monomers are repeated many higher times, while some monomers have been repeated lesser times. So they form higher order repeats, and the satellite DNAs, which are different in constitution, they occupy the peripheral position, whereas the similar satellite DNAs are whereas the similar satellite DNAs are present at the central region of the centromere, and satellite DNAs differ, the monomers differ in their constitution of nucleotide. That is, the sequence of nucleotides does not remain entirely the same. They differ, but they constitute a monomer of about 171 nucleotide, and that's how they have been classified as alpha family. Human satellite 2 and 3 are also present there. So that was all about this structure, structural specification. And one more thing, a peculiar type of centromere is found in many organisms, that is holocentric, that entire chromosome having the centromere. And it has been said that this holocentricity, origin of holocentricity, has led to the as an adaptation. It has originated as an adaptation to certain specific features in their life cycle. Say, for example, phytophagous insects, they face clastogens from the plants. So plants secrete clastogens which can destroy the chromosomes of the phytophagous insects. So it had been said that the holocentricity in insects had developed as an adaptation to the clastogen secretion by the plants. That is, they are the clastogenic adaptation of the insects to the plants, to the defense of the plants. Certain other insects face segregation, and which leads to the breakdown of their chromosomes. So such insects have also developed holocentric chromosomes. Certain kinetoplastids have also developed holocentric centromere. And so the holocentricity had been quite diverse and it has originated perhaps as a convergent evolution from the point centromere, and our scientists have come to the conclusion. One more thing, a peculiarity of holocentricity is that the holocentric insects in the holocentric centromeres in insects have lost CENPA or its variants entirely. So up till now, we were defining centromere by the presence of CENPA. But here, there is an exception that is found in case of holocentric chromosomes of insects, where the CENPA had been entirely lost. How has it been lost? It is said that when the architecture of centromere was being changed from monocentric to holocentric, somehow they dispense with the CNPA, and the role of the CNPA is being played by some other proteins, as had been reported from some fungi and some from kinetoplastids. Now, the next section deals with the epigenetic specification of the centromere. So we have up till now studied the genetics in the light of the sequence of nucleotides on the DNA. Mutation involves change in the sequence of nucleotides, and that mutation leads to the change of sequence of nucleotides in the RNA and proteins ultimately. So it was the nucleotide sequence which was sacrosanct, which was of prime importance, which should not change. Of course, it is of prime importance and it should not change. But there are many other factors which are above and over genetics. Those factors also influence the expression of genes, and these factors are acetylation, sumylation, methylation of side chains, and also methylation of certain nitrogen bases too. And by these addition or deletion of these groups, they can have impact upon the expression of DNA, such as acetylation leads to the expression of DNA in a better way, whereas methylation leads to the suppression of DNA. So the DNA activity can be modulated. The expression of DNA process can be modulated even without any change in the nucleotide sequences, and those factors which influences the DNA activity without any change in the nucleotide sequence are called epigenetic factors, and epigenetic factors have to play a big role in the specification of centromeres, right from the CNPA, that is centromeric protein A, and its variants, to the methylation of lysine at certain points of histone, canonical histones, such as methylation at 0.9 of canonical histone 3 will lead to the compaction. And if pericentric chromosome also is hypomethylated, it will affect the centromeric function. So the methylation of the centromeric nucleosomes, the side chains of the histones at the centromere, particularly the histone 3 and histone 4, H3 and H4, at H4 at the 28th point and H3 9th point. So the methylation will lead to the compaction, and compaction will lead to the suppression, and the suppression of will impart heterogenetic characteristic to the centromeric chromosomes, because they will not be expressed easily. Since they have to provide integrity to the point of attachment, they will be closely attached, they will be tightly bound, and they will get attached to the spindle microtubules on either side, and while maintaining the integrity, they will get separated to the either poles. But my dear students, it is not that the centromere kinetochore attachment point is a passive point of attachment for the centromeres, for the spindle fibers. It is not a passive docking point. It is not a passive point of attachment and maintaining a strength of the chromosome. Rather, it is a very active point. It regulates the entire process of segregation of chromosome by seeing to it that the chromosomes have been properly arranged and they have been attached on either side by the microtubules, and that's how we have seen in the present class that how a spindle assembly complex senses that the tension is not there, and it activates the MMC mitotic sorry it activates the MMC, and that activation of MMC will inhibit the anaphase chromatic complex, the APC, and the inhibition of anaphase chromatic complex will stall the process of inhibition, stall the process of segregation of the chromosomes to the different poles. And this process will remain stalled till the chromosomes are properly arranged bi-orientationally at the equator of the dividing cell and get attached on either side by the centromere, get attached to the centromere or the kinetochore, centromere kinetochore complex on either side, so that the centromere kinetochore can sense tension that is being built up by the attachment of the centromere, attachment of the spindle fibers, microtubules on either side and being pulled apart. If this tension is felt, then the spindle assembly complex feels that there is everything okay and the cell division should proceed now. And so it is activated, and its activation leads to the activation of MMC, and MMC, which in other situation had activated the APC, that is anaphase promoting complex, will give green signal to anaphase promoting complex. Or in other words, you can say that inhibition on the anaphase promoting complex will be withdrawn because MMC has got inactivated as the APC is not activated because the tension is being felt by the centromere kinetochore complex on either side. And this inactivation of APC, sorry, inactivation of spindle assembly complex and then MMC will activate the APC oblique cyclosome, that is anaphase promoting complex, and this anaphase promoting complex activation will give a go-ahead signal to the segregating chromosomes, and these chromosomes, since it had been ensured that they get properly attached on either side to the centromere, they will move towards the poles, and each of the two poles will have the complete set of the chromosome, the entire complement of the chromosome, and that's how the two daughter cells that have emanated from one mother cell will have the entire set of the chromosomes. Each of the two will have the entire set of chromosomes, and that is how the fidelity is maintained. And we can say that, well, the segregation has taken place in a fidel way. So that was the story which I could sum up in a few lines and which I have detailed in so many classes. So you might say that I was over stressing upon my points, but my intention was that you get proper explanation for each of the diagrams and each of the highlighted points that I have added in my slides. Now, to conclude, I can say that centromere is a highly conserved region. You see that there is centromere down the line, right from plants to we human beings. And they are performing the same functions. So they are highly conserved, but at the same time, the centromeres are highly evolving. So what a paradox. The centromeres are conserved because they perform a particular role in the cell division, and each and every cell has to divide except for the minuscule exceptions. So they have to divide, and in their division, the centromere has to play a particular role. And that role I have described in detail. Still, the centromere is evolving fast, and it is the centromere, evolution of the centromere, that imparts the sexual isolation and speciation. And this process of evolution had been well studied in the evolution of horses, where the shifting of centromere has taken place. And I have also explained the silencing of the centromere and development of the neocentromere. But these processes may be fatal for a particular generation, but across the generations, these processes have led to the speciation and contributed in the evolution too. So I'm leaving you with that paradox that it is highly conserved as well as the highly evolving region of our genome, that is the centromere. And although I have solved so many paradoxes regarding the centromere structure, its genetic specification, its epigenetic specification, its role in the segregation of the chromosomes, its role in the maintaining the proper segregation, that is the fidelity of segregation, still I am leaving you with a paradox that is the most conserved region is the most evolving region too, and that you can imagine the importance of centromere in our genome. So that is the highlighting point of the centromere that I'm leaving you with. So thank you very much. Thank you for being with me for so long. And it was a pleasure for me to deliver this lecture. Any confusion, any gray area that you may feel, you should correlate with me by posting your questions in the comment box or directly contacting me so that I could explain them in my future lectures too. So thank you. Thank you very much. Nice meeting you.